Checkpoint question 9. What effect would a chemical that blocks the voltage-gated sodium channels in the excitable axon membrane of a neuron have on its ability to depolarize? Well, it's the sodium channels that have to open to lead to depolarization, so it would make it more difficult for the neuron to depolarize. Number 10, what effect would decreasing the concentration of extracellular potassium have on the membrane potential of a neuron? So one thing that we know is that the concentration of sodium outside the membrane has to be high, and the concentration of potassium has to be high inside. So if the concentration of potassium needs to be high inside and sodium high outside, if you lower the concentration of potassium outside, that's going to make the outside less positive. So the inside, I'm going to have to come back to that one. So go down to number 12. 2 axons are tested for propagation velocities or speeds. One carries action potentials at 50 meters per second, the other at 1 meter per second. Which axon is myelinated? And of course it's going to be the first one with the faster action potential. So going back to question 10, I had to pause and think about it and think about how to explain it. So we already know that the concentration of potassium in the extracellular fluid or outside the cell, we already know that that concentration is low. It's already low. So if we decrease the concentration of potassium even lower outside the cell, then what's going to happen is it's going to lead to even more potassium leaving, because potassium, there are leak channels for potassium that are open all the time, and potassium is always going to diffuse down its concentration gradient from inside the cell where it's high to outside where it's low. So if you make that concentration gradient even greater by lowering the potassium outside the cell even more, then more potassium is going to leave. So what does that do to the voltage across the membrane when potassium leaves? We already said it makes it more negative, right? Well, if it's at resting now, making it more negative than resting is going to it's going to be called hyperpolarization. So it's going to hyperpolarize the membrane. All right, moving on, the synapse, and luckily we've already covered the synapse in chapter 7 because we had to when we went through the information on the neuromuscular junction. So the synapse is defined as the site where the neuron communicates with another cell. And it's specifically on the neuron, it's going to be where the axon terminal of the neuron communicates with another cell by coming very close to it and releasing neurotransmitters that are going to bind with receptors on the target cell. So information is transferred through the release of chemical messengers called neurotransmitters. We learned about one neurotransmitter called acetylcholine. And we learned about the neuromuscular junction, but there's also neuroneuronal junctions. That's not in the slide anywhere, but the junction is between, it's neuro something. It's between a neuron and its target. So if the target is a muscle, it's called a neuromuscular junction. If the target is a gland, we call it a neuroglandular junction. If the target's another neuron, we call it a neuroneuronal junction. So in a synapse, you always have two cells that are involved. You have the presynaptic neuron, always. And then you have the postsynaptic cell. If the postsynaptic cell is a muscle, it would be the postsynaptic target cell. But in this case, we're looking at a synapse between two neurons. So we've got the presynaptic neuron. This is the axon terminal of the presynaptic neuron. And then this is the plasma membrane of the postsynaptic neuron. And it can be anywhere along that neuron. It can be the plasma membrane of a dendrite or a cell body, or it can even be another axon. You can have axonal connections, synapses. But anyway, here's what we've got. We've got a presynaptic neuron which holds the neurotransmitters and releases them across the synaptic cleft. That's the gap in between the neuron and its target. And then we have the postsynaptic neuron that has receptors for those neurotransmitters. In chapter 7, our target cell, it would have been the motor end plate of a skeletal muscle fiber that had receptors for acetylcholine. So in this case, it's just the postsynaptic target cell is another neuron, but it still has receptors for neurotransmitters. So one that we've already learned about is acetylcholine, or ACH. It activates cholinergic synapses, and here's the steps. I just want to remind you of step 3. Acetylcholine binds to receptors on the target cell and triggers depolarization of the postsynaptic membrane. So it depolarized the motor end plate of our skeletal muscle fiber. So we know that acetylcholine is a neurotransmitter that can be used to activate, to cause skeletal muscle contractions. But there are other neurotransmitters, and on this page I just want you to see the names, and then we'll talk about what their effects are. There's norepinephrine, or noradrenaline, means the same thing. Epinephrine and adrenaline are the same thing. There's dopamine, GABA, and serotonin. And if you've had any experience with medications, taking any medications for depression or anxiety or other mental conditions, these are the neurotransmitters that those types of drugs will affect. It will be levels of norepinephrine, levels of dopamine, levels of serotonin. Prozac is probably the most common one of these medications, but there's a lot of them that affect levels of serotonin. So there are 2 other neurotransmitters that are actually gases. They're not chemically, they're not like the other neurotransmitters, but there's nitric oxide and carbon monoxide. These are gases that act as neurotransmitters. They will bind to the receptors for neurotransmitters and cause similar effects. So what we need to talk about are the effects of the neurotransmitters. The only type we've learned so far has been excitatory. So an excitatory effect means that it leads to an action potential, leads to depolarization, and then an action potential in the target cell. That's what we saw happen with acetylcholine at the neuromuscular junction. It caused contraction of the muscle. So that's excitatory, but we also have neurotransmitters that are inhibitory. So if the effects are inhibitory, they're going to cause hyperpolarization and inhibit the action potential instead of leading to an action potential. They're going to lead to not having an action potential. So most of the time, your acetylcholine and norepinephrine are excitatory. They trigger depolarization, so they have excitatory effects, usually. So it's not always the neurotransmitter. Sometimes it's the type of receptor that it binds to. But most of the time, acetylcholine and norepinephrine are excitatory. Dopamine, GABA, and serotonin tend to be inhibitory. They trigger hyperpolarization. But you can have different effects from the same neurotransmitter. Acetylcholine can be inhibitory depending on the receptor it binds to. So what's really going to determine if it's excitatory or inhibitory is not only the type of neurotransmitter, but the receptor that it binds to. Checkpoint question. What effect would blocking calcium channels at a cholinergic synapse have on synapse function? Now, because of the wording, you may not recognize this question as being something that we actually have experience with this in chapter 7 in the muscle chapter. The cholinergic synapse is the neuromuscular junction. That's a cholinergic synapse. Acetylcholine binds to receptors on the motor end plate of skeletal muscle. So if we block the calcium channels, if you remember, calcium channel is what led to the release of the neurotransmitter. So if we block those calcium channels, we're not going to have an action potential, basically. Now, we're going to move on and talk about the structure of the brain and the spinal cord, and we're going to focus more on the brain. So before we talk about the actual brain, we're going to talk about the meninges, which are the layers outside the brain and the spinal cord. The meninges, there are 3 layers, and their function is stability, protection, fluid flow, and interaction with blood vessels. There's the dura mater, the arachnoid, and the pia mater. They surround the brain and the spinal cord. The cranial meninges are continuous with the spinal meninges, as well as continuous with the nerves. So there are three layers. Dura mater is on the outside, is the outside layer. The arachnoid is on the inside, and the pia mater, the arachnoid's in the middle. The pia mater lies the inside part, so actually contacts the surface of the brain. I did want to mention the subarachnoid space, simply because that is the place where you would get an epidural for pain, you know. So it is deep to the arachnoid epithelial layer, so it's within the arachnoid mater. And it's arachnid means spider, right? So spiders make webs. So the subarachnoid space is created by a web of collagen and elastic fibers, and it's filled with CSF, or cerebrospinal fluid. And that's where you can, that's how you can receive the epidural there, and you can also take, do a spinal tap, you know, and get cerebrospinal fluid for doing testing on it. Okay, now the segments of the spinal cord, and like I said, I'm skipping some of this information because it's just way too much, but there's 31 total segments that give rise to the spinal nerves. The segments are kind of similar to the segments that you have in the vertebrae. You have actually 8 cervical, whereas you only have 7 cervical vertebrae. You've got 8 cervical spinal nerves. You have 12 thoracic spinal nerves, just like vertebrae. You have 5 lumbar spinal nerves, just like the vertebrae. And you have 5 sacral and 1 coccygeal. Each spinal segment, which corresponds to like the 31 different segments on the spinal cord. So each segment has a pair of spinal ganglia. Which is right here, we often call this the dorsal root ganglion. And the ganglia contains the cell bodies of sensory neurons. So the dorsal root, or posterior root, of the spinal segment, of the spinal nerve, is going to contain axons of sensory neurons. And the ventral root, or anterior root, is going to contain axons of motor neurons. So this is going to be motor neurons, and this is going to be sensory neurons. So that's why we call the spinal nerves mixed nerves, because they contain both sensory and motor neurons, axons of both of those. All right, the gray matter of the spinal cord is made up of glial cells and neuron cell bodies. Remember, the neuron cell bodies contain Niesel bodies that cause the gray color. And the white matter is white because myelin is fatty. It's composed of lipids, and lipids are white. So the white matter contains myelinated and some unmyelinated axons in the spinal cord. All right, checkpoint question. Damage to which root of a spinal nerve would interfere with motor function? And that would be the ventral root, because the dorsal root is sensory. A person with polio has lost the use of his leg muscles. In which area of his spinal cord could you locate the polio-infected motor neurons? I have to look this up, but leg muscles. Which area controls leg muscles? Okay, I read that question harder than what they were making it. They're really just asking where in the spinal cord do you have the motor axons of the motor nerves, and that's going to be, the motor is going to be the ventral root ganglion. But as far as the specific region of the spinal cord, it will either be in the lumbar plexus or the sacral plexus. And this is further on in the notes, so I'm going to go back to where we were. Way back to where we were. Okay, so where could you locate the motor neurons that control the leg muscles, basically? And that would be either in the lumbar or the sacral plexus of the spinal cord, and it would be in the ventral root of the nerve that you're studying. Okay, so we're going to start in the next lecture with the brain, and we'll go into a lot more detail with the brain because the brain is so fascinating, and I want you to know more about the brain really than the spinal cord.